It's Thursday. It must be time for molecular genetics. Hi, y'all. All right. I have been thinking, but I don't have a lab thingamabob quite set up yet. Partly, I haven't given you enough information and thinking and things to think about to be able to really do some of the PCR-based DNA sequence stuff we're doing and we want to do in the lab. We're getting to it. Hey, give yourself a break, because believe me, I gotta give myself a break. Otherwise, I make myself crazy. Where's my coffee? Hold on a second. Now, so we're talking about some of the historical parts of molecular genetics and exactly where we are in the modern era of molecular biology. Genes are, in fact, we now know our DNA, and that we know the dogma of information, that is transcription. DNA will serve as a template. RNA is transcribed from the DNA, and then translation. Proteins are translated using the RNA as a template. But in terms of the technology of gene cloning and how we can begin to actually use the tools of molecular genetics, we drove across a couple of interesting points last time we spoke. Specifically, we were talking about DNA's ability to go from being double-stranded to single-stranded. And this is denaturation. And then we can have the strands of DNA come back together again to reanneal, and this is known as renaturation or hybridization. Now, what we were mentioning was the idea of TM. And we know that A T base pairs melt at a lower temperature than G C base pairs. So depending on the ATGC concentrations, the ratios, we can actually, there's a formula that you can find where you can give the sequence size, because to a degree the amount of DNA plays into this, but probably once you get above 40 or 50 nucleotides, the length of the DNA isn't as critical. The critical points are going to be the ATGC composition. What the heck? I'll run up there. So we got what's typically known as the GC content. This is a critical variable for TM. The next critical variable is going to be the salt concentration. That is, if we've got a low salt solution, that is going to tend to favor exact base pairing and not allow for base pair mismatch. Another way of saying this is it is going to raise up or actually it'll lower the melting point. Lower salt is going to have lower stability, but let's not worry about that right now. Another part of determining TM is the ability in an experiment to add specific kinds of destabilizers. A classic destabilizing agent is something known as formamide. Formamide is able to destabilize the DNA interactions and make it so a DNA sequence that would normally have a TM, say, of 80, now might have a TM of 42. And you're going to say, why is that important? And it's because of the way we do an experiment, and I'll tell you. So these are some of the things that you figure into your calculation of what the melting temperature is going to be, the melting point. The fourth and potentially most interesting is going to be the degree of base pair mismatch. So, do you have an exact sequence match, or do you have some kind of what we might think of as a hybrid, or some kind of non-exact sequence match? So what this means is that TM is dependent on conditions. TM is not a fixed characteristic. It is contingent upon a set of known conditions. So with any kind of sequence or experiment, you have to begin looking at the GC content, the salt concentration that you're going to run the experiment in, the formamide concentration that you may or may not choose to use, and the degree of base pair mismatch that you're either looking for or you're willing to tolerate, and that can determine what your TM is. Now, what this means is we can carry out experiments in which we are hybridizing DNA sequences together in two ways. We can have what we think of as high stringency, and we have at this little to no base pair mismatch. So if you're looking for exact sequences, you would carry out the experiment under conditions that are very near the TM. On the other hand, a really cool experiment might be to run this at low stringency. Low stringency means that you're going to allow for base pair mismatch. How much base pair mismatch? Glad you asked. How low can you go? Well, let's face it. If you're looking at sequence and you probably are getting somewhere around 25-30% base pair mismatch, at that point you probably are looking at garbage, and you probably don't have a sequence that is particularly related to the original sequence you're doing, and so your experiment is worthless. Go ahead and just pick a random piece of DNA sequence or just invent something and you're fine. So stringency, if you get so low, you're basically just pulling out garbage, and you have to adjust it because you don't want to pull out garbage, but you don't necessarily want to have exact matches. Now, why is this? Because let us imagine that we've got a gene that we identified in a fruit fly. We want to find the homolog to that gene in humans. Now, we know it's not going to be an exact match, but we can use the fly gene in a low stringency screen to identify the human homolog. What we're going to do is talk about a couple of different experiments using stringency that are probably not things that you're ever going to be exposed to or are being done anymore, and they probably haven't been done. The last time I did these kinds of experiments would be probably around 2000 or so. So we now pretty much use PCR for these kinds of experiments. We'll talk about stringency issues in PCR in a bit. But for now, let us travel back in time to the mid-90s. All right, were any of you even born then? I don't want to know. Fine, I was a postdoc. This was my profession. This is what I did. The truth is, back in like 93, I was hot, man. I was doing high-tech stuff. Not so much now. No! I'm doing things. It's all good. Here's the experiment. And it's actually a really cool idea and cool experiment. And that you have to recall back in the late 80s, mid 80s, 90s, people began first cloning genes from complex organisms particularly. the idea that a gene found in a fly would have a homologue in a vertebrate wasn't something that was completely expected or understood. Particularly because there's some very cool genes in flies that are developmentally regulated and are actually generating the basic body plan. They are sort of master switches for turning on a range of different genes for, like, say, eye development. And to think that they may actually be in humans was a huge deal, because now you can begin applying the kind of model system approach to looking at humans by working on flies. So here's an example. A guy named Matthew Scott had identified a really interesting developmentally regulated gene that was known to actually shift the fate of different cell types. It's what he called a homeotic gene. And in flies, and we'll be talking about this later in the semester, but in flies, there are a series of mutations that can convert one body part into another body part. And this is known as homeosis, it's Latin stuff. In vertebrates, homeotic mutations aren't very common. There are apparently some homeotic mutations that can occur in your spine, in which one vertebra will actually be structured like the vertebra below it. And I'm not sure if that causes you back problems or what, but it is something that is a way we can think about it. In flies, it's rather dramatic. There are homeotic mutations. One is known as antennapedia, in which instead of having antenna on your head, those antenna get converted into legs. There is proboscopedia, the mouth parts. Instead of having mouth parts, they have spongy mouth parts. They get converted into legs. Legs seem to be the thing. There is another homeotic mutation known as ultrabithorax, in which the flies have a set of wings, and then they've got a set of little stubs. They're called halteres, and these are little paddles that they probably use, or they're used aerodynamically to tip the body, kind of like the way helicopters fly. And in dipterans, which flies are, they've got one set of wings and one set of these little nubs, these little halteres, beep, 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 and then these great big wings. Ultrabithorax can change that set of nubs into a full set of wings. And all of a sudden, you have a fly that instead of having two wings, or one pair, now has two. It looks like a dragonfly. And in fact, that's how dragonflies have two sets, because they have this mutation in ultrabithorax. So Matt Scott at University of Colorado Boulder had been doing some work on cloning genes in flies. He'd come across and cloned one of the first homeotic genes. And one of the tools that you use to analyze genes and genomes is something that's known as a DNA gel blot. And this was invented by a guy named Southern. And so the term has now been, this kind of gel blot is referred to as a Southern blot. I have to tell you that while Mr. Southern, Dr. Southern, great man, never met him, but whatever, I choose not to use those trivial terms, because his name Southern led to the idea of, let's call other gel blots by directions. And so you have a northern blot and you have a western blot. And while this is all very amusing, they are terms that, while I do use them, I typically try not to. And this is my own personal opinion. Very prestigious scientists who were, or a guy that was one of the first to really develop some of the tools of molecular biology, a guy named Eric Davidson. And we talk about some of his experiments later in the semester. A brilliant guy at Caltech. He never referred to those experiments as northern blots or southern blots. He always referred to them as a DNA gel blot or an RNA gel blot. And it's part of this acronym crap that we need to get away from. And so I tend not to talk about southern blots or northern blots. And I don't mean to be rude, but I don't think highly of people that do. That was an opinion. Sorry I made it. I'm embarrassed now. Everybody's fine. You can use whatever term you want. It's just not in front of me. Okay. We're going to talk about a DNA gel blot, a southern blot. Here's how it goes. You take genomic DNA. You isolate genomic DNA from, let's say you have a fly gene, isolate genomic DNA from a fly. You're going to cut this up using a restriction enzyme. Now, we're going to be getting into restriction enzymes when we start talking cloning. Hopefully in genetics we talked about it, but maybe not. A restriction enzyme, they're all from bacteria. A restriction enzyme is an enzyme that recognizes a specific DNA sequence and cuts at that spot. For example, there is an enzyme from E. coli known as EcoR1. And it was one of the very first restriction enzymes that was purified. It recognizes the site GAATTC. And it cuts in an asymmetric fashion like that. Now, if you notice, this is a palindrome, CTTAAG, and it's going to cut at this spot. So you end up with what is known as a complementary base pair overhang. But that's not so important for right now. Regardless, we got restriction enzymes that cut at these specific spots. And this is a 6 nucleotide restriction site or recognition site. So it's generally referred to in the parlance as a 6-hitter. There are a bunch of restriction enzymes that recognize 4 nucleotides. There are a few restriction enzymes that recognize 8 nucleotides, and then there are just a handful that recognize a restriction site that's like 11 or 12 nucleotides. The bigger the restriction recognition site is, the less frequent it's going to be found in a genome. So 4 hitters, they're found all over the place. 6 hitters, they occur probably every 1,000 or so nucleotides on average. There are 8 hitters that might occur maybe every 10 to 15,000, and then there are a variety of enzymes that there's probably only 1 restriction site in the entire human genome for some of these more esoteric guys. So the point being, you take genomic DNA and you cut it with the restriction enzyme, and you're going to get a range of DNA fragments. So now, what we can do with a genomic DNA blot is begin to look at the genome architecture and looking to see how or where the restriction sites are in this gene, and a variety of different kinds of gene organization, exon, intron kinds of things. You can figure it out. So it's a worthwhile experiment being done back in the late 80s, early 90s. And it works like this. You take your genomic DNA, you carry out an exhaustive, thorough restriction digest, meaning you carry it, let it sit at 37 degrees for probably 5 or 6 hours. You purify it if you need to, and you run it out on a gel, on an agarose gel. So here is our agarose gel. And there's the wells. And you electrophoresis this thing. And so now you're going to see just an absolute smear of DNA in the lane. You're not going to distinguish individual bands. It's going to be a ton of stuff. And you take a picture of it, great. You stain it with ethidium bromide like we do. Now, which I think about this in Molecular Cell, that's where we ended our labs, was running the PCR gels, and we never got any further than that. We'll talk. But regardless, you stain the gel. Now what you're going to do is actually transfer this DNA onto a sheet of either charged nylon membrane or nitrocellulose, because the DNA is actually embedded in the gel and you can't get at it. So you set up a blot. And in order to set up a blot, there are a couple of ways you can do it. you take the gel and you lay it on top of this nylon or nitrocellulose sheet, and it's just a little bit of very delicate sort of membrane material. And typically what you would do, and you would find this in lots of different molecular biology labs, is you'd set up a dish of a salt solution buffer. And then you had some kind of little block or stand that you'd put in here. And you'd put the gel here, or actually you lay a sheet of filter paper, like Wattman filter paper, on the gel. And so it's wicking up. You then put a, hold on. I'm thinking about this because I stopped doing it this way, and I'll show you my other way of doing it, really quite clever. You put the membrane on. There's the membrane. And, wait. Oh, phooey. All right, hold on. Put the gel down. Then you put your membrane down. And now you have a big stack of paper towels. This is why molecular genetics labs are always flying through paper towels. So you got paper towels, membrane, gel, and then essentially a wick. And so what's happening is this wick is pulling this saline solution up. The saline solution is being drawn through the gel by the paper towel. The DNA will be pulled along by capillary action or by diffusion and bind to the membrane. And you set this up and let it go overnight. This is your gel blot. Now, just to say, the reason I was getting backwards is that this was the way that it was originally designed. And I'm looking over here at my set of books, and a set of the molecular cloning lab manual from Cold Spring Harbor. It became sort of the Bible for everybody. And this is the way to do it. Now, what I started doing and realized is a lot easier is rather than setting the blot up like this, you do it backwards, where you get a big stack of paper towels, put the membrane down, put the gel down, and then take a sponge soaked in the saline solution, plop that on top, and you pull it down. You don't have to have this big bucket of solution. So that's the standard way, though. Okay? So now, at the next day, the gel is actually compacted. The paper towels are soaked, and when you disassemble this whole thing, you end up with a membrane that is an exact duplicate of your gel. So, this now is our membrane. Okay, what we're going to get set up to do now is a hybridization experiment. And the first thing we have to do is render the DNA on this gel blot single-stranded. And the way we do that is expose it to base. So we're going to soak this thing in 1 molar sodium hydroxide. What 1 molar sodium hydroxide is going to do is cause the DNA to absolutely open up and separate. Now, I used to worry, but hey, if it's on this blot, isn't it right next to each other? Nah, it pulls it apart, and they don't re-anneal on the paper very well. So you soak it in 1 molar sodium hydroxide. Now, just as a little point, after I started doing this a bit, I realized, hey, I could use a sponge, soak the sponge in sodium hydroxide, and denature the whole thing while I'm transferring it, and that's really the cool way to do it. But regardless, you soak it in sodium hydroxide for 5 or 10 minutes. Then you neutralize it and wash it a few times. Then the next thing you have to do is bake the membrane to fix the DNA onto the membrane, and that keeps it actually now from re-annealing on the membrane, which I'm not sure that it would, but regardless, we always bake it. You can also UV cross-link it. You now have this cooked membrane. So now the membrane is single-stranded, or it has single-stranded DNA in it. It's ready for a hybridization experiment. Okay, the next thing we're going to do is make a hybridization probe. And a hybridization probe is a DNA fragment that you're using as the probe for hybridizing to a band here. And back again in the old days, the standard way to make a DNA hybridization probe was to label it with P32, radioactive nucleotide. So you take typically deoxy-CTP, cytosine triphosphate. And just as a little note, when we're talking about the phosphates, we talk about the fact that you've got the 3 phosphates. I'm not sure if it's 03 or 04 here. You don't care, and neither do I. We got this structure. Since we're using a deoxy, it's missing that oxygen. Now, you got 3 phosphates here. You got to make sure that when you order it from the manufacturer, New England Nuclear, that you have it with the appropriate phosphate is radioactive. And there's a very clever little nomenclature. It is the alpha phosphate, the beta phosphate, and is that gamma? The gamma phosphate. All right. Now, when you're linking together nucleotides, a very specific reaction is occurring in which these 2 phosphates are being removed. And you're left with this phosphate as part of the phosphodiester link. So you want to make sure in this experiment that this is the radioactively labeled phosphate. And what is released is the 2 phosphates. Now, why they don't call this a diphosphate, I don't know, but it took me a while to realize when I started hearing this term what they're talking about. The 2 phosphates are referred to as pyrophosphate. We start talking about DNA sequencing. There is a what was called next-gen sequencing, but it's pretty old-fashioned now, called pyrophosphate sequencing, is that you can observe or detect a nucleotide being attached to the chain by the release of the pyrophosphate, these 2 phosphates. Regardless, we've got the alpha phosphate is labeled. Now, nobody sells and nobody uses beta phosphate being labeled. If you're doing a kinase reaction where you want to look at the transfer of a phosphate through some kind of kinase, you would use gamma phosphate. Now we're looking to have DNA that's labeled, and so we're going to use the alpha phosphate. So we take our DNA fragment, our gene piece of interest, and we label it up. The standard way is to use DNA polymerase, put in the different nucleotides, and add radioactive DCTP, allow the DNA polymerase to begin polymerizing and replicating DNA. We won't need to get into the details of this, but all kinds of different enzymes can be used. And after a couple hours, you purify this fragment, and what you have made is new DNA that incorporates the radioactive label. Now, you typically have to purify the DNA because there's a lot of the label that won't go into your probe. And this is where having the little Geiger counters that we used to have on our bench was nice, is that we generally would purify the probe by doing a little bit of gel filtration, which separates out small molecules from big molecules. The DNA being pretty darn big, it flies right through the column. The P32 that's not been incorporated, or the DCTP that hasn't been incorporated, is going to stay up in the column material. And you could use a little column and actually let it drip. It's very convenient to actually set it up in a centrifuge, like a little clinical centrifuge. And you put it in a syringe, run the DNA through it, and your probe would come out in the solution. You'd have about a couple hundred microliters of labeled probe, and you hold the Geiger counter up to that, and it should really go off. And then you hold the Geiger counter up to your syringe that has the unincorporated nukes, and if things went well, it won't be going anything. We used to have these little yellow Geiger counters that had a limiter switch that if it got too high, it would just start to squeal. And so if you had a screamer, beep, you knew you were good. So you can generate a piece of DNA that is now radioactively labeled. You take that DNA, this is called the probe, and first you want to remember to denature the probe. And that's generally done by boiling it for 5 minutes or so. You could add a little bit of sodium hydroxide and then neutralize it, but that's a pain. It's a lot easier just to throw it in a temp block and let it boil for 5 minutes, and you're ready to go. You then add this to your hybe juice, your hybridization mix. Hybe juice, or the hybe mix, is going to have a reasonably high salt concentration. It has, of course, a buffer. There's a solution known as saline sodium citrate, or also known as SSC, and that typically is what you generate your hybe mix in. Often, or most time, also you'll have things like some kind of protein mix. It could very easily be bovine serum albumin or just powdered milk, non-fat dried milk. A wonderful lab at Baylor, they actually published a paper in Biotechniques on using Bailey's Irish Cream as their protein mix, and their point was you can then later use the remaining Irish cream to celebrate your victories or to soothe your defeats. So you then often will have some kind of non-specific DNA, non-labeled, like salmon DNA. Salmon sperm DNA was used a lot, I don't know why. And this is mostly just to kind of coat the membrane in non-labeled DNA to block non-specific sticking, non-specific binding of your probe. So you do a pre-hybe where you wash the blot with this stuff, and you do that for an hour or so, and then before you go home for the evening, you add probe. So now you're going to add the P32 labeled probe. So this is radioactive as heck, and one of the standard tools was to put the membrane into a seal-a-meal bag, cut the corner off, seal it all up, cut the corner, put your pre-hybe mix in there, let that go, and then you seal it back up and let it go. And then before you go home, you cut the corner again with your little scissors and shoot in your radioactive probe and squish it around real good after you seal it up. It is radioactive as heck. And you allow this to carry out its hybridization. So, hybridization. What we're doing now is allowing these two single-stranded molecules, the probe and the single-stranded molecule in the gel, to interact. Now, you want to carry this specific hybridization reaction out at a very low TM, because you don't want to mess around with, or at a low temperature, well below the TM. You don't want to have the chance of your hybridization mix not binding. You want it to bind. Now, the lower the TM, or I guess the lower the temperature, the more likely you are to have non-specific base pair mismatch. But at this point, you don't care. So what you would typically do is carry out the actual hybridization generally around 60 degrees. Now, 60 degrees is rather warm. What is that in Fahrenheit? It's probably about 140 Fahrenheit. Regardless, I don't know and I don't care. And so we would have these water baths that could be run up to pretty high temperature. But it gets steamy, the water can evaporate. So one of the nice things you can do is you can add 50% formamide, and that is destabilizing things such that the equivalent to 60 degrees now becomes 42 degrees, which is a much more legitimate temperature. Most water baths can do that. You don't have to worry about it evaporating overnight because it's not near boiling or anything. And so you typically would carry it out at 42 degrees. All right. Now, we're going to go ahead and allow this to hybridize overnight. And you come in the next day, and you can save the probe, and you can re-denature it and use it again. P32 has a half-life of about 2 weeks, and so as long as you do it within a reasonably short period of time, you can use the same probe a couple of times. So if you were to take this gel and expose it to X-ray film after hybridization, you would have one great big black blob. I mean, it is radioactive all over. So now what we're going to do is what's called the stringency washes. So we begin washing. And let's just, for the sake of our experiment, say we've got the fly gene and we're going against fly DNA. So we can go for very high stringency. That is, we're going to select for only exact base pairing, and anything else is going to melt off. So we start washing. And just the general way you do it is you start out with a pretty high salt solution and running it at 68 degrees and let it wash, or 60 degrees or so, 65 degrees, and let it wash. Pour that off into the radioactive waste because it's going to be really hot. All that non-specific stuff just washes right on off. And you then begin the actual stringency washes. So now you've got a low salt, high temp wash. Low salt and high temp, that means that you're going to be favoring those hybridizations, or those hybrids that are near exact matches. And so you can do this for a couple of hours, wash it a few times, and when you use the Geiger counter on this thing, it's going to be almost silent, and hopefully you'll hear some ticking over here, a couple of spots. You take this gel and you put it onto a sheet of X-ray film and let it develop overnight. You come back and develop the film, and what you're going to see is this. Instead of having all of those bands, you're going to see one band light up. One or two bands. That is the gene in its genomic location in whatever EcoR1 restriction fragment it happens to be on. It's there. Now, that is a standard experiment that you might do just to confirm that yes, everything's fine. This piece of DNA that you cloned is in fact in the organism. Because all kinds of weird things can happen. The next thing you would do is carry out not a high stringency hybridization, but a low stringency hybridization. So let's see what that would look like. You're going to run the gel the exact same way, but now when you get to your washes, rather than doing a low salt, high temp, you do, let's just say, a moderate salt and a moderate temp. And this is going to be favoring base pair mismatch, because now you have set up conditions in which you won't melt away exact matches because you're not anywhere near the TM, and you won't melt away base pair mismatch. And so what you can do is sort of calculate up, but there's also just the general rule of thumb, but you can calculate up. This is what the TM is going to be, and then you might go 15 degrees below the TM. That would be the salt concentration and the temperature that would give you 15 degrees below the TM. And now what you might see is this band really strong, and then a few other bands. And what these are indicative of is other genes in the genome that share sequence similarity. Now, the really cool breakthrough that came with doing low stringency hybridizations was something that Matt Scott called a zoo blot. Because what he did is go to the Denver Zoo and got little bits of tissue from all the different vertebrates they had. He had some gorilla, he had giraffe, he had elephant, he had rhino, I don't know what the heck he had. He had kangaroos, they had mice, they had humans. They ran restriction digests on isolated DNA from each one of these samples, ran it out in a gel, and carried out a low stringency hybridization. Now, in flies, you expect to see the same kind of pattern. But then in gorilla and elephant, who knows what we're going to see? Lo and behold, it turned out that gorilla had some bands, some that were pretty strong. Elephant also had a set of bands. And what he has discovered is that there are genes within these different vertebrate genomes that share sequence homology. Now, this is a critical experiment for being able to identify homologous genes in other species, a low stringency screen. In the last moment here, we do typically do PCR these days rather than blots. We can carry out low stringency PCR as well. And so I'm going to just throw this out. We're going to get back into this on Tuesday. We'll start talking about PCR specifically and getting a little bit more of labby bits. But for now, when we're talking about the polymerase chain reaction, PCR, and I'm kind of going through this because it is assumed that everybody's had molecular cell and is familiar with the technology of PCR, because even in our molecular cell class last semester, we did run PCR. Remember, what it is you got genomic DNA, and then we have a set of very small 20 or 30 nucleotide sequences that are known as primers. And they're going to bind to a region of the genomic DNA such that they direct DNA polymerization going that way and DNA polymerization going that way. So they flank the region of homology, or the region of interest. And the primers are designed to be complementary to the two strands of DNA, so that they'll bind in this way. And when you're doing PCR, as we say, you have to have a bit of DNA sequence information. Now, if you're going to be doing human DNA and you want to amplify a known gene, you go to the Human Genome Project, you look up your gene, you identify the sequences, and you go, done. And the manufacturer of the primer, they'll do a quick calculation, and they will tell you what the TM for your primer sequence is. And so it might be 73 degrees. And they'll determine it based on the sequence and using a standard salt solution that is sort of the PCR primer buffer, and that they can give you that. All right. What you're going to do when you run a PCR reaction is typically carry out the annealing step at 10 degrees below the TM. That's standard. So that would be 63 degrees. We can carry out a low stringency PCR, though, by allowing the annealing temperature to be maybe 20 degrees, 25 or 30 degrees below the TM. I try not to get much cooler than like 37 or 40, because really at that point you're going to be biting into just all sorts of crapola. But it does work. And so we can do an equivalent low stringency screen by having the portion of the PCR reaction that we call the annealing temp. We can have it greater than 10 degrees below the TM. All right, that's all I got to say for now. It occurs to me that we probably need to go through PCR fairly specifically, but we'll pick on that up on Tuesday and start getting into some experimental design and see what we can get into. That's all I know. Thank you for your attention. Rock on.